This episode is brought to you by SkinCeuticals, the #1 medical aesthetic skincare brand worldwide and the leader in antioxidant science. To learn more, visit www.skinceuticals.com.
Hello and welcome to Skincare Confidential, the podcast supporting the Science of Skincare Summit, an annual educational conference occurring in Austin, Texas in 2024, November 8th through the 10th. I am Dr. Ted Lane, your host for today, and it is my great pleasure to introduce and welcome Dr. Mark Birch-Machin, a professor at the Newcastle University Institute of Cellular Medicine. He is a professor of molecular dermatology. Dr. Birch-Machin, thank you very much for joining us.
Great to be here today.
Great. Is it okay if I call you Mark for the rest of the episode?
Absolutely.
Okay, great. Please call me Ted.
Okay.
All right. So Mark, so great to have you. Thank you. We are really going to focus on, you've done a plethora of research, but we're going to focus on your research that revolves around the effect of UV and visible light on mitochondrial DNA.
Sure.
Let's start with what is mitochondrial DNA, because I'm not sure that many people really understand what this is.
Sure, and actually, when you start mentioning mitochondria, if people don't come across it before, their eyes glaze over, they feel a little bit sleepy. So I always step back and say, actually, the mitochondria are the batteries of the cell. They're the power packs that produce the energy that makes everything go round in the cell. And so really, if you think about mitochondria as your batteries and whether you want to keep the batteries topped up or stop them running down, then that's a good starting place. Okay,
great. So they're the engines of the cell, the batteries of the cell.
Yeah.
So they produce the energy, and the energy for the cell is ATP, right?
Correct.
Okay. And therefore, they are intimately and inextricably involved in cellular respiration.
Yes. And so actually, mitochondria are involved in all energetic processes. They actually are found in every cell of the body. I first started working in neurodegenerative disease, so muscle and brain, and now I'm working in skin. But mitochondria are in every cell of the body, and so essentially, any tissue that requires energy, ATP, mitochondria are at the center of it all.
Okay, great. So absolutely integral to the cell's functions.
Yeah.
And there's this mitochondrial DNA that you're really focusing on, which is an interesting DNA, right? Because it's different than the genetic material that we commonly think about in the nuclei of our cells. This one, which of course is derived from your both sets of your mother and your father. The mitochondrial DNA is different, though, isn't it, Mark? Can you go through that with us?
Yeah. So the DNA we get from our mother and father is what we call the nuclear DNA. All right. Mm-hmm. That's much bigger than mitochondrial DNA. And mitochondrial DNA, by comparison, is much smaller. But actually, because of where it is and what it does, and it's at the kind of the fulcrum of the bioenergetic process, it has a kind of almost like an overinflated role compared to what its DNA is. And so unlike the nuclear DNA, which we get from a mother and father, the DNA housed inside the batteries of our cells actually is inherited from our mothers. So it's maternally inherited only because of various processes where the mitochondria from the father is kind of tagged with a kind of like a death protein, as it were, and so that gets kind of mashed up. So actually, it's maternally inherited, and also the interesting thing about it is the fact that it's not protected by a lot of proteins like histones. So essentially, it's naked DNA. All right, DNA with its clothes off, as it were, if for want of a better word, naked DNA. So because of that, it is very vulnerable to damage and stress in the cell, however that stress may present itself.
Oxidative stress is what we're going to focus on. And so just to reiterate, so this is DNA that is important, integral for the mitochondrial functioning. It doesn't have access to the nucleotide repair enzymes, all of not all, but many of the enzymes that we commonly attribute repair in our nuclear DNA. The mitochondrial DNA does not have the advantage of being exposed to. Really.
Correct. And so, for example, normally, you know, if you have damage in your nuclear DNA, which is also, you know, which is protected by these histones and things like that. So let's say if it gets through the histones and you get DNA damage in your nuclear DNA, like creating a crime scene, so to speak, the nuclear DNA is very efficiently repaired. So the crime scene is tidied up. All right. So you can go in and see if a cell has been stressed or under trauma in nuclear DNA. And because it's such a good repair mechanism, there are damages, and obviously that we know that. But for the large part, that damage is repaired. In the mitochondria, they are lacking certain repair mechanisms or are inefficient at normal repair mechanisms. And so because of that, you get an accumulation of damage. For example, there's a disorder called xeroderma pigmentosum, right? Which in dermatology is where people with XP, and in America, I think children with XP are called moon children because they only go out at night, all right? Because actually, if they go out during the day, they suffer a potential risk of 2,000 to 3,000-fold increase in skin cancer because they can't repair their sun-induced DNA damage, right? Right. So what is interesting, so those are XP patients. What is interesting is in the mitochondria, they do not have the same repair mechanism. They do not have nucleotide excision repair. And so because of that, the mitochondria can't repair their sun-induced DNA damage either. And actually, because mitochondria and all of us, then actually we have kind of almost many examples of XP existing in our skin of every individual.
Interesting. And so does the mitochondrial DNA therefore carry these mutations, and could that be a sign of aging? Could you effectively determine the aging of the skin based on mitochondrial DNA damage because it's not being repaired?
Yes. And so there's actually, before I answer that question, your listeners might be thinking, "Oh my goodness, I got many examples of XP in my skin all the time," or actually in all your.
Yes, let's avoid mass hysteria, Mark.
Okay, yes. Let's avoid mass hysteria now. Okay, because basically what happens is there's something called complementation within mitochondrial DNA. All right. Okay. So because so in your nuclear genes, in your mother and dad, there's two copies, two alleles. All right. In mitochondria, you have hundreds and thousands of copies, depending on which tissue you have. All right. So muscle and brain would have thousands, skin would have hundreds, for example. And so because of that, what that means is you have complementation. In other words, the undamaged mitochondria help out the damaged mitochondria. And so essentially what happens is your cells can tolerate quite a lot of damage in your mitochondria before it begins to affect cellular function. All right, which actually from a marker point of view, and we will probably come on to that later, is very useful. But actually, I just want to, that should stave off mass hysteria. All right, because so actually, mitochondria are very good at tolerating its own damage because the kind of undamaged ones. It's a very kind of generous thing, isn't it? The fact that the ones that are undamaged helps out the ones that are damaged, and wouldn't that be nice across the world in all societies? But so it's very, very nice from that point of view. You alluded to this thing about aging. Now we know that aging, there's many factors to do with aging. There's telomeres and senescence and all this kind of stuff that goes on there. And actually, the reason why mitochondria have begun to take kind of quite a major role in aging is because there's a theory called the mitochondrial theory of aging, and at the center of that is what's called the vicious cycle. Okay. And essentially, you've got three components to the vicious cycle, one of which is mitochondrial DNA, which we've mentioned. The other is the function of the mitochondria, which is the function of the battery. And the third component is the oxidative stress, which you've also mentioned as well. And so they exist in a little triangle, all right? And so essentially what it means is the fact that if mitochondria are sitting there minding their own business, they're unprotected, they don't have these histones. They have inefficient repair mechanisms. So they accumulate DNA. They accumulate DNA damage, which affects the function of the battery. So that function is compromised. And then because the function is compromised, it's almost like the exhaust fumes of an engine, that what happens is they pump out more reactive oxygen species, which increases oxidative stress,
Right.
which in itself then damages more mitochondrial DNA, affects mitochondrial function, leads to more exhaust fumes, more oxidative stress. And you get this kind of runaway vicious cycle of aging. And so the challenge there is to move us from the fast lane of aging to the slow lane of aging by interrupting and slowing that vicious cycle down.
Okay. And is that vicious cycle related to this term inflammaging, where we have the inflammation associated with aging?
Yeah. So all of this is related because actually, part of inflammation process is this increase in oxidative stress, and there's a kind of key trigger aspect to that. And the reason why mitochondria are involved in that, and if you draw a Venn diagram, there would be an overlap there, is the fact that two things. I mean, lots of things happen when we age, all right? So I'm simplifying this dramatically, all right? From a mitochondrial-centric point of view. Okay. So, but when you look in the mirror, as it were, deeper down, two things happening when you age. With mitochondria, your batteries tend to run down. Okay, and we know that, the way we make ATP, the amount of ATP we make declines with age. Okay. And what tends to increase is the fact that oxidative stress increases. Now, the mitochondria produce 90% of cellular oxidative stress by what's called a cascade system. It's like knocking over a domino. You knock over one thing and it leads to a cascade. And so that's why there's a link with that inflammaging.
This episode is brought to you by SkinCeuticals, the #1 medical aesthetic skincare brand worldwide and the leader in antioxidant science. To learn more, visit www.skinceuticals.com.
Let's focus on the oxidative stress and how to prevent it or mitigate it as much as we can. You've done some research to show that both UV and visible light affect the mitochondrial DNA, right?
So we haven't done visible light affecting mitochondrial DNA, but we've done UV and, we've actually done visible with infrared. Okay, so we haven't been able to tease out the infrared, but what we did is, we kind of divided sunlight or solar light into different components, the UV and then visible light and infrared. Yeah. Okay.
And can you tell us the results of this research in terms of, obviously we would imagine the UV light affects change, cyclobutene pyrimidine dimers, CPDs, that occur in the mitochondrial DNA like they occur in the nuclear DNA. But what about the visible and infrared light that you've researched?
So this was a publication that was probably about 6 years ago that we published, and we're still working on some of the implications of that. But very simply, what we found, we asked the question. There's been a lot of reports about the role of UV, and we know UVA, UVB has a profound effect on cellular damage. And so we used mitochondrial DNA as well as nuclear DNA, and we measured reactive oxygen species as well as markers of that damage, almost like parameters of the cell trauma. This crime scene that we've been talking about, the fact that mitochondria aren't very good at tidying up the crime scene, so it's still there. So we hit some skin cells with UV, right. But also what we did is we using various filters, using a solar simulator, which simulates sunlight. We then added back the visible and the infrared component of sunlight in the study. All right. Okay. So we couldn't actually separate out at the time, although we are able to now, visible light and infrared. So and what we found was the fact that separately, the visible and the infrared really didn't do an awful lot under the conditions that we used. Not to say that they don't have an effect, but under the conditions that we used in sunlight, they didn't seem to have much effect on the parameters. The parameters being nuclear DNA damage, mitochondrial DNA damage, and reactive oxygen stress. Okay. But actually, when you combine it with UV, it almost poises, it almost enhances the visible light and infrared effect, which tells us the fact that on their own they may not have so much of an effect, but actually when the cell is, as it were, damaged or troubled by UV, their activity is enhanced profoundly by, you know, mechanism, the fact that you've actually got some stress happening already. And then when you add some different stress, then that enhances that. And so what we found is, so there's a difference there. And interestingly, we found that was a difference between the fibroblasts, which are in the dermis, and the keratinocytes in the epidermis. What we found is that actually this enhancing effect was really only observed in the dermal fibroblasts and not in the epidermal keratinocytes. And so it tells us the fact that different layers of the skin will react differently to different wavelengths of light, which you intuitively feel anyway, because you know penetration. But actually there'll be something different about dermal fibroblasts dealing with these different stresses. And so because of that, that has implications in terms of aging, protection, everything else. Yeah.
That's fascinating. So when you're referring to the different wavelengths, as we know, the UVA, the longer wavelength, dives deeper into the skin, it penetrates deeper into the skin, hence probably affecting the fibroblasts more than UVB, for example.
Yeah.
Right? Yeah. Okay. So there's this nefarious synergy that happens with visible and UV light to enhance the production of reactive oxygen species and therefore damage. Now, in your research, did you include any kind of protection, antioxidant protection, sunscreen protection, anything like that?
Yeah, so so within that study we didn't. So that study that was published in FASEB. We didn't actually include any protection or anything like that. It was just a question of what happens when you traumatize cells in this way. And you know, the conclusion of that is there'll be different chromophores for infrared, visible, and UV in those cells. And I guess the aspect in terms of protection is if you find out what those chromophores are, then you can actually design intelligent protection for them. Sure. Okay. And so, but with the tools that we have, and then we're actually able to look at using sun protection products to actually see whether or not they can help to protect against UV-induced mitochondrial DNA damage, because you know the nice system that we have is the fact that you know we know UV damages mitochondrial DNA very directly. We don't understand the mechanism of infrared and visible, so in terms of you've got to start somewhere. So let's start with UV, and that's part of the study that we've done recently that we're going to present at the EADV this coming week, actually.
Ah, wonderful. Okay, can you talk about the study, or do you want to wait until you present it?
Well, I think we can talk about it, because obviously it's produced as an abstract at the conference. So it's obviously it's probably even public domain now. So I think we can talk about that, because it's actually been submitted and accepted as a poster presentation at the EADV. Okay. So within the study, we haven't actually we actually worked with SkinCeuticals, and the work that was done was completed by our spin-out company called Skin Life Analytics, which is a Newcastle University spin-out company. In other words, what happened is the fact that it's not similar to most universities. Sure. You allow that technology that you have as an academic to be spun into a spin-out company in exchange for, shares and all that kind of stuff and things like that. So it just means the fact that we can actually take a lot of the clever stuff that we do and then actually help to make a difference in the cosmetic world, etc.
Commercialize it so it can be used.
Commercialize it. That's right. So it actually puts it into the hand of more people.
Understood.
Which is great. So actually the work that was done was obviously between SkinCeuticals and Skin Life Analytics.
Okay. And
what we did was we asked the question whether or not these sun protection products, which were at SPF 50, could actually prevent the UV-induced mitochondrial DNA damage.
Now, in both keratinocytes and fibroblasts.
No, we just did it in fibroblasts. The only reason,
okay.
the only reason we didn't do keratinocytes is because with keratinocytes, the best model is actually using primary keratinocytes. So you get a lot of donor variability. So you have to do it loads and loads of times, and so it kind of makes the system a bit noisy. So actually, by using human dermal fibroblasts, you can actually get cell lines which actually make the whole thing, reproducible, and what you're going to get every time. So the only change that you see will be due to the intervention, and the intervention at this point is obviously plus or minus the sun protection product.
Okay, and what we learned from your research that was published 6 years ago is that these dermal fibroblasts are much more sensitive to visible plus UV light than the keratinocytes. So it makes sense to focus on the fibroblasts as well from that perspective.
Absolutely, absolutely. So there's a lot of sense in that. There's a lot of traction with that study. And so because we've done a lot of work on, doses and everything else, what we did was we took our human dermal fibroblasts and we irradiated them with effectively 2 standard erythemal doses. And so the difference between standard erythemal dose and minimal erythemal dose is because MEDs are take into consideration skin types, whereas SEDs don't. It's a very much a kind of a pure medical physics. And because I've been kind of brought up by medical physics, they don't allow me to talk about MEDs. So so I have to talk about SEDs.
If I could just interject. So a minimal erythema dose is based on your skin type, right? It's the amount of UV that will cause redness in the skin. The erythema is redness, right? So
exactly.
Everybody has their own MED. And what you're saying is that would be very difficult to do in vitro. And so instead you use a standard dose that will cause the damage that you're looking for.
Perfect. Yes. And then actually 2 SEDs is roughly equivalent to about 20 to 40 minutes sun exposure in the Mediterranean sun. All right.
Okay.
So it's a very physiological dose. It's not, you're not looking at something which is hours and hours. So
if I can interject, just clinically, 20 to 40 minutes outside is pretty much what we tell people to get weekly for vitamin D conversion. So this is not outside of the norm of what dermatologists commonly recommend, quite frankly. So very interesting. Okay. I can't wait to hear the results.
But actually within that context, and I've been involved in many vitamin D discussions, etc., because obviously we need exposure to get vitamin D. Obviously this is clearly in single cell monolayers rather than multi-layer skin. And so you can get more sun exposure in multi-layer skin without actually being traumatized by what I'm about to tell you.
Yeah, good. Well, thank God our fibroblasts are not directly exposed to the sun on any given day. So
exactly. And there's reason for that. And there's reason the fact that those fibroblasts are below the sternum corneum, below all these other cells. And so because of that, and there's good reason for it. And so what we did is we irradiated those cells, and we did our proper positive and negative controls. The negative control is obviously covered up, and the positive control is fully exposed. And also we included a sham control, which is basically chassis with no SPF. All right. So essentially what we found with those positive and negative controls was the fact that we got our maximum damage and our minimum damage. All right. So everything worked from a technical point of view. And then you do the same experiment, and you actually have sun protection products, and there were 4 of them, suspended between the light source, the UV light source, and the underlying skin cells, right? So if you can imagine a kind of 3-way thing, you've got the light source up above, you've got the sun protection products and a solid support, which is UV transparent below, and below that you've got the skin cells.
Okay.
And then having irradiated those cells, what you then have to do is you then have to extract the DNA from those cells. And then assay using our proprietary technology, which has actually taken many years to, we've had to walk around the whole genome and everything else to find out that what the mitochondrial genome is sun specific or sun sensitive, right?
Interesting. Okay.
So we use those proprietary primers to ask the question, do the sun protection products lead to decreased damage, or none at all, or even increased damage? You've got to have every 3 options available to you.
Right.
And so what we found in all 4 instances was the fact that the sun protection products allowed a protection in all cases, up to about a 320% protection, or 3.2-fold protection, compared to when it's not there. So basically, so they're doing a very good job, and that's really based on the mitochondrial DNA damage. And what we do is we do our appropriate controls. We actually have loading controls. We actually allow for a mitochondrial loading gene as well, which is well away from the damaged area. So we do all our appropriate controls. What we did find, because I guess the next question would be, okay, Mark, of these 4 products, is there a winner there? Is there a front runner?
That was my question.
Yeah, the front runner. If I was a betting man, it was a horse race. Which is the winner?
Which one do I use, Mark? Which one do I use?
Well, actually, because they're all at SPF 50 and containing, because they afford that protection, there were differences between them, but no significant differences between them. So all 4 showed a significant difference between, maximum sun exposed, and so there were a highly statistical difference. I think they were all less than 0.002, significant. So very highly significant. But when you actually assay analyze the differences between the products, even though by eye there are slight differences, statistically there's no difference at all between the 4.
Thanks, Mark. And can you please go through the different ingredients that were included in the 4 sunscreens that were tested in the study?
Yeah, so the 4 sunscreens that were tested were top-selling SkinCeuticals sunscreens, and both mineral and organic sunscreens were used in those products. And it also included a variety of filters, either US or international filters, due to the regulation requirements based on the formula. But we know the fact that we only put on about 40 to 60% thickness on a good day.
Right, because
the amount of
put on, at least an ounce of sunscreen on normal
sun
exposed areas. Yeah.
So nobody really puts on 2 milligrams per centimeter squared, which is actually what the SPF is tested at, right? Okay, even if you're meant to put on 2 milligrams per centimeter squared, I would challenge anyone to put it on regularly. And so actually, the idea of actually going for a higher SPF means you probably, because of inadequate application, as it were, or patchy application, you actually end up with the protection that you would probably want to have. Does that make sense? Rather than if you go for 30, you end up with less. And so actually, going for 50 is quite prudent from the experimental point of view, because that's probably what you might suggest that you might start with.
Well, I can tell you that mirrors what dermatologists say every day. Which is use the highest SPF you can find, because you're probably not putting it on correctly anyway.
Yeah, absolutely. And as I say, we talk about this a lot, and within the discipline, even though what 2 milligrams per centimeter squared looks like, I just don't think people would put that on a regular basis. You might do when you're thinking about it, but not when you're rushing and things like that. Yeah.
So as we kind of end our time here, and it's gone by so quickly as it always does, what are your thoughts now about this? You mentioned this a little bit, but it's something that I've thought a lot about and continue to as we consider the applications of the hallmarks of aging to the aging science and inflammatory skin disease as well. And your thoughts on kind of turning away from clinical signs and symptoms as endpoints towards more molecular endpoints as we look at, as you mentioned, DNA protection instead of an SPF for sunscreen. I think as we look at a lot of the skincare products that we use, we're seeing the skincare industry move away from just using clinical endpoints, clarity, pigmentation, fine lines and wrinkles. And now we're moving towards more, DNA expression, RNA expression. What are the molecular endpoints? DNA methylation. You can go on and on. Do you think that we're at the age now where we should start seeing that more and more with not only SPF, but also, the more common skincare products that are being introduced now?
Yes, I think what it does is it the findings that we with the abstract and the poster that we're presenting at the EADV with SkinCeuticals and Skin Life Analytics, with the technology, as it were, spun out from Newcastle University, there are clearly, looking at the literature, there are, very good molecular markers that reflect the damage not only caused by UV, but pollution, lifestyle, diet, etc. And so it's a very much a holistic aspect. And so really, part of the kind of strap line of the spin out we set up was to make the invisible visible. And I think that's the question you're asking, isn't it? It's almost like, these deeper down invisible things that are going on. And one of our invisible things is mitochondrial DNA damage, but there are other invisible things that are going on as well. And so you want to make them visible to us so that we can do something about skin protection, repair, all those kind of things before we see the visible signs that we're all familiar with, those wrinkles, fine lines, uneven skin tone. So we want to do something about it at earlier stage than actually once it becomes visible in terms of, skin imaging, etc. And we use skin imaging machines as well. So it's not like I'm saying they're not good. They are.
Like confocal microscopy or...
Exactly. You can, clearly what you can do is you can use these kind of deeper molecular markers, the invisible things, to actually see whether or not an intervention works. And actually, if with mitochondrial DNA and these other markers giving you an objective measure, you can tell whether or not what you're intervening with or your lifestyle is making that damage more or less. And so you can manage your lifestyle because you can see whether something's going up and down. With nuclear genes, you've got it. You have a propensity, you have a risk ratio of getting something, not getting something. With things like mitochondrial DNA, because it's so sensitive, it can go up and down depending on the stress to your cell. And so because of that, you've got a way of tracking how your damage is going with your lifestyle, and then working with the interventions that are available. And, some protection products clearly are of a very effective measure, whether or not you're applying it properly, if it's working properly, et cetera, et cetera. So I think it's very exciting what you could bootstrap the new markers to things that we're familiar with, like SPF, because that's what we're familiar with. But if we can, it's like we can actually bootstrap it to something else, and then actually we can maybe mature those protection factors to other parameters.
I mean, if you can, the analogy here to me is this constant glucose monitoring that we use in diabetic patients, for example, right? Where they have constant feedback on the control of their condition. The idea, if we apply that to skin aging, perhaps it's skin scrapings that we send in once a month or once every year to a lab such as yours to do the molecular, analysis of aging. And that could tell us, okay, it's time for you to start using this product or change this in your diet or change this in your lifestyle, for example. So I think as we, progress in the science, Mark, I hope you agree with this. I think it's time for us to start considering what we do in other diseases or states is something that we could do to aging as well. We need to treat it like aging to me is a disease that we can treat. Yeah,
exactly. And actually we've, within the same spin out company, within the work we've talked about with the EADV, it's like a contract research lab, it's like we're there to test products and actually, but also what we've got is this skin scraping that you've talked about. We can actually take a skin swab. We developed a skin swab where we take there's enough skin cells on that skin swab, and it's taken years to develop from a biopsy to a swab, and actually then analyze your mitochondrial DNA damage at that time point. And so that allows us, if you say your first swab is time 0, then you have an intervention or a change in your lifestyle or putting on a product. Then you take that swab at, 6 weeks or 12 weeks. Then you can figure out actually how is your DNA damage in your mitochondria going, which obviously is reflecting the health of your skin.
This is brilliant. Okay. Well, we've run out of time, unfortunately, but we could go just into that subject again. So I'd love to get you back just to talk about that and send me one of those swabs. I want to do this. So
the deal.
All right, great. So this has been Skincare Confidential. My name is Dr. Ted Lane. I'm a board-certified dermatologist. I have had just the wonderful pleasure of interviewing Dr. Mark Birch-Machin, who is the professor of molecular dermatology at the Newcastle University Institute of Cellular Medicine, who has done some really groundbreaking and incredible research on the effect of sun protection on mitochondrial DNA in fibroblasts, and really the tip of the iceberg of everything that we could talk about, Mark. But thank you so much for your time.
Thank you, Ted.
All right. Thanks, everybody. Don't forget to give us a like, subscribe, and refer us to your friends. And thank you very much.